So I'm really pleased to be able to give you an update on recent studies on HIV transmission and the viruses that are involved in this process. As Monty Python said, now for something completely different. I think it's important to remember that different routes of infection are accompanied by different risks of transmission. And heterosexual transmission being the least frequent, somewhere in the order of 1 in 200 to 1 in 3,000. MSM interrectal about 10 times more frequent, risk of 10 times more, and MTC even higher. Intravenous depends, of course, on the dose, with transfusions being nearly 100%, while intravenous drug users are somewhere greater than 1 in 100. The route not only influences risk, but it also influences the pressures put on the virus during transmission. And so I think when we compare different studies, it's important to realize that different cohorts may have different risk factors that influence the nature of the virus that's being transmitted. This slide shows in red the viral load early in infection, peaking somewhere in the order of 20 days, and then plateauing out to what we call set point viral load. And the colored bars are defining different stages that Fiebig was able to establish studying plasma donors, with stage 1 being the first stage where we could see virus in the blood, that is, where viral RNA can be detected. Stage 2 being where we can detect the viral core antigen through an ELISA, the P24 ELISA. And then stages 3 through 6, different phases of antibody specificities and reactivities. Really, for the most part, what we've been restricted to in our larger cohort studies have been phases 2 through 3, although we can sometimes go back and find phase 1 samples. But it's usually phase 2 through 4 that we've been analyzing the virus. And we got to realize that the virus we're looking at is about 2 to 4 weeks after the initial infection event. And we're extrapolating back to understand what virus initiated that process. What's been very clear now since the early 90s with papers from Walensky and Wolfs is that this diverse population of viruses, this genetically diverse population of viruses that we see in the transmitting partner that I'll frequently refer to as the donor, is contracted down genetically. It undergoes a genetic bottleneck during transmission, so that 80 to 90% of infections are established by a single virus variant from the donor, and we call this virus now the transmitted founder virus. It's the virus that establishes infection, and I think it's important to point that out because we do now believe that more than one virus may be transmitted to the uninfected recipient, but it's the one that actually establishes infection that we're most concerned about. Now, in some instances, depending on inflammation or ulceration or effects of STIs, more than one virus variant from the transmitting partner can be transmitted. And in fact, in IDUs, we see multiple viruses sometimes being transmitted, although even there we see a genetic bottleneck occurring. I'm just showing here a phylogenetic tree of a transmission pair from a couple in Zambia. Phylogenetic trees define the genetic diversity of a virus. And what you can see on the lower part of the screen here are the transmitting partners viruses, each of these amplified from a single virus. And the horizontal lines, the length of the horizontal lines, define the genetic distance between one virus and another. So you have to go all the way back and back to here in order to get a sense of how distinct those viruses are. And you can see that the donor viruses, or the transmitting viruses, virus partners' population of viruses, is quite diverse genetically. In contrast, shown in blue, are the viruses from the newly infected recipient, or the newly acutely infected individual. This is about 20 days after infection, and you can see that the viruses have virtually no horizontal distance and in fact are homogeneous. It's a clonal population of viruses derived from a single branch of the phylogenetic tree, indicating a single genetic variant was transmitted. Now, it's not always possible to have transmission pairs, and in a really landmark study, Brandon Keel employed modeling and phylogenetic analysis of single genome amplified sequences from viruses isolated very early from greater than 100 acutely infected individuals to impute the transmitted founder virus, and showed in this large group of individuals that 80% of the infections were the result of a single virus variant. Transmission involves both stochastic and selective components. Obviously, chance has to play a big role. The transmitted variant's got to find itself in the genital tract for sexual transmission. If we're talking about sexual transmission, it's got to be located in the genital tract. It's got to interact. It's got to actually manage to find its way to the genital or rectal mucosa. It's got to cross that epithelial barrier and find and infect a susceptible target cell. And it has to have a sufficient number of secondary target cells for infection to spread and establish a localized infection that can then become systemic. This slide shows a sort of a current concept of HIV infection of the female genital tract. It's actually derived from studies in rhesus macaques by Ashley Haas and his colleagues, Chris Miller, for example, and it's inferred from a series of timed analyses. The virus obviously has to get across either stratified or columnar epithelial cells and find a CD4 T cell. That CD4 T cell produces virus and induces the influx of activated target CD4 T cells that can amplify the signal and allow virus then to spread to the efferent lymph nodes and become systemic. The key feature is the virus has to find its way into the gut-associated lymphoid tissue, which is where the massive depletion of CD4 cells occurs. So this process, modeled from monkeys, and more recent studies argue that sometimes the virus actually can get out of the mucosal tissue very quickly and is not restricted entirely by this process. So I think that's perhaps going to depend on the particular animal or person infected or the virus infecting them. I've mentioned the chance aspects of transmission, but there's also clearly a role for selection. We've known, again, for many years that a majority of infections are initiated by viruses that use the CCR5 co-receptor. This molecule, in concert with CD4, allows changes in the viral surface glycoproteins, these structures on the surface of the virus, to allow the virus to fuse into cells. There's a second receptor, CXCR4, that late in infection in subtype B in particular, the virus can switch to, but in transmission, this virus is the one that's selected. In addition, several studies have shown that the envelope glycoproteins of transmitted founder viruses have fewer glycosylation sites and shorter variable loops, which is where a lot of the glycosylation sites are located. That means there's less sugar modification on the surface proteins of the virus. And in addition, it's likely that viruses must target CD4 T cells that express the gut homing marker alpha-4 beta-7 to efficiently establish infection. Jim Arthos has shown that transmitted founder viruses have a much better affinity for alpha-4 beta-7 than viruses isolated from later in infection. And in a recent study by Reddy sorry Sid, I put your first name up here instead, by Reddy and Ansari, has shown that by treating animals, pre-treating animals with an alpha-4 beta-7 antibody, you can delay and in fact prevent infection by SIV and dramatically protect the gut-associated lymphoid tissue from infection and depletion. So alpha-4 beta-7 cells appear to be the carriers of systemic infection. Even in the genital tract, where the genetic makeup of genital tract HIV is reflective of that in the blood, and where we can see enrichment of genital tract specific populations, for example, shown schematically here in this cartoon by these red viruses that are far more dominant. What we've seen is systemic infection with a variant that is relatively minor in both the genital and blood virus populations. And that variant sometimes is more similar to genital tract viruses and sometimes more similar to viruses we isolate from the blood. So if we have enough transmission pairs, we should be able to apply a probabilistic model to really try to understand what are the selective pressures on the virus and how are they are being applied. And Jonathan Carlson at Microsoft Research has been actively involved in these studies. Basically, it says if you've got two variants in your virus populations that are there with some relative frequency, and in this case I'm showing one virus at 25% and the other virus at 75% of the population. Is transmission just a probability of that frequency, or is it relative to or is it a function of transmissibility? In the first case, one might expect that 25% of the time the minor variant would be transmitted, whereas if there's transmissibility of the virus involved, we might see a greater fraction of those viruses with the minor, a greater fraction of infections with the minor variant. So we wanted to test this experimentally. So we first asked, well, are amino acids present in 3 functional proteins of the virus, GAG, POL, and NEF? Are any of those amino acids preferentially transmitted in the transmitted founder virus relative to ones that are present in the transmitting partner viruses? So first we determined the most common amino acids at each position in each of these 3 proteins for viruses in a Zambian cohort of 375 people. We performed 454 deep sequencing of the genome for 5 transmission pairs, analyzed the frequency of each amino acid at each position in the donor and recipient viruses, and then calculated the transmission frequency of each consensus amino acid and each non-consensus amino acid. What this slide shows is on the x-axis the frequency of any amino acid in the donor quasi-species, in the viruses that had the opportunity to infect. And on the y-axis, the frequency with which those amino acids were transmitted. And you can see that for consensus amino acids, that's a relatively straight line. And in fact, it's pretty much a direct correlation between the frequency in the population and the frequency of transmission. However, when we look at non-consensus polymorphisms or non-consensus amino acids, the whole curve is shifted down significantly so that, so that if we were to look at amino acids present 85% of the time in the population, those are transmitted pretty much with that frequency for consensus amino acids, but less than 70% for non-consensus amino acids, showing a clear selection bias against non-consensus amino acids and what we think is against less fit viruses in the population. We applied this then to all 137 transmission pairs that we'd accumulated in the Zambia cohort. And again, if you're looking at female to male transmission, The curve looks very similar. It's a linear relationship between frequency now in the cohort as a whole of an amino acid and frequency of transmission. It's very different when we're looking at male to female transmission, shown in blue here. The curve is shifted to the left, and in fact, from a 50% frequency point on, the curve is essentially flat, saying that less frequent amino acids, the ones that one might consider to be mutations selected for by the immune system, are now being transmitted with equal efficiency, and therefore the virus transmitted is going to be less fit. Surprisingly, when we looked at men with genital ulcers and inflammation, we saw a similarly low barrier to transmission. So it says that there must be some barrier at the mucosa that is imparting this selection, particularly in female to male transmission. So what this tells us is that HIV transmission involves competition between viruses. Ultimately, one virus has the ability to replicate faster than is necessary to generate a localized population that can then spread systemically. Whereas other viruses may never get out of the mucosa, may just potter along, but never actually expand to the point for getting out. And others just may die out because they become defective or are defective. But competition is clearly playing a role here between the different viruses. It's likely that competition also plays a role following exit from the mucosa. And a recent study by the US military HIV research program, where they've been sampling individuals, generally sex workers, at twice weekly intervals, and being able to catch the upswing of virus very early in infection. They've been able to sample now both in Phoebic stage 1 all the way through to Phoebic stage 4. And this is a study called RV217. And what you can see here in this particular individual, participant 40061, is that at 7 days after infection, estimated date of infection, there's a major variant being transmitted, but at a very almost undetectable level is a minor variant. 25 days, at day 25 in contrast, the minor variant has now become the predominant virus in the population, and the major variant has decreased significantly. But this minor variant is only then replaced again by the major variant. So there's obviously ongoing selective pressures that cause, in instances where more than one virus is transmitted, changing pressures on the viruses to which they respond differently. Now, can we translate this evidence for selection into identifiable biological traits? Because such traits obviously could provide clues for targeted interventions, particularly vaccines or other biomedical approaches. And recent studies from the Chauhan lab, published in 2013, compared not just single gene products of the virus. There'd been quite a lot of an extensive analysis of the envelope glycoprotein of transmitted viruses to their non-transmitted counterparts. But for the first time, this group compared transmitted founder virus infectious molecular clones from chronically infected individuals to those from acutely infected individuals, which they identified as transmitted founder viruses. What that study showed was that transmitted founder viruses replicated better in CD4 T cells, and also that those viruses were more resistant to the antiviral effects of interferon alpha. But this was most evident in studies of subtype B viruses. And that's shown here. If one takes what I'm showing here is in gray viruses growing in activated CD4 T cells in the absence of interferon. The solid line being the transmitted founder viruses. This is a sum of a group of, I think, somewhere in the order of 20. And the dotted line, the circulating chronic circulating virus, IMCs. And then below in black are the effect of interferon on those viruses. And you can see that the chronically circulating viruses are impacted much greater. For subtype B, that's really obvious when one samples the amount of virus replication at day 6, day 9, and day 12, where at day 9 there's almost a 2-log difference in the ability of transmitted founder viruses to replicate relative to the chronic circulating viruses in the presence of interferon. But this was really not the case for subtype C. In the case of subtype C, you can see that the transmitted founder and chronic circulating viruses were very similar. And because in the paper by Fenton May, it was shown that viruses replicated in the presence of interferon better as transmitted founders, but 6 months later had a much greater sensitivity to interferon. The initial attribution was that maybe there was a difference in the chronic circulating viruses rather than the transmitted founders themselves. And in fact, if you look at this graph, you can see that the main difference is in the sensitivity of the circulating viruses. We wanted to look at this in more detail using our Zambia transmission pairs. And these are derived from HIV discordant couples that were followed over a number of years. They were counseled and tested, and that results in a 2/3 reduction, in fact greater than 2/3 reduction in transmission in these individuals. And that has translated into the clinic when this program has been rolled out into government clinics. So one might say that if one is going for 90, 90, 90, maybe we should have 90% of people knowing the status of both themselves and their partner in the future. But some infections do occur, about somewhere between a quarter and a third, and about 80% of those are what we call epidemiologically linked. The transmission occurred within this discordant couple partnership. So we identified female to male and male to female transmission pairs within Phy-big stage 1 and 2, so very early, either just RNA positive or just P24 positive, this stage of acute infection. We amplified full-length genomes from individual virus RNAs and sequenced over 150 genomes from 6 transmission pairs. You can see a phylogenetic tree of those shown here. In each case, a single genetic variant was transmitted, and that's shown in blue. It's probably a little difficult for you to see that. And in each case, again, the transmitting partner's virus was quite diverse. Using very high fidelity polymerase and some novel cloning methodologies, we've been able to actually clone directly from PCR amplicons. And that's shown in the black arrowheads at the ends of some of these virus genotypes. And so we've been able to also use these amplicons to generate authentic full-length IMCs. But first we asked, what about this selection for consensus? If we look now at these full-length genomes, does this recapitulate what we've seen with GAG, POL, and NEF in our larger cohort? And the answer was yes. You can see in each case the blue virus is underneath the median for the non-transmitted transmitting partner virus genomes that we amplified, confirming the selection bias, even in this very small number of transmission pairs that we'd seen for GAG, POL, NEF sequences. We were then able to take a series of generally 3 to 5 infectious molecular clones from the transmitting partner and the transmitted founder virus and compare them for replication. And that's shown in this slide here. The bars, the height of the bars indicates the ability of the virus to replicate in activated CD4 T cells. And what you can see is that while the transmitted founder virus in blue is clearly in most cases in the upper quartile of replicative capacity. It's rarely the most highly replicating virus, and in some instances actually is a rather poor replicator compared to the non-transmitted viruses. We then looked at interferon sensitivity, again in a subset of these, and what you can see is that there is really no consistent evidence of higher resistance in these transmission pairs, these subtype C transmission pairs. You can see a couple of instances where the transmitted founder virus grows better in the presence of interferon than in the absence of interferon compared to the non-transmitted virus. But in other instances, it's worse, and there's a large range that we're seeing here. So I think that the impact of interferon in this case, and I should say that we included in these studies 3 transmission founder 6-month pairs that had been reported in the Fenton-May paper. And again, there we were able to reproduce that the transmitted founder viruses were in fact replicating much better in the presence of interferon than the 6-month viruses. So, transmission is characterized by a severe genetic bottleneck that can be modulated by genital inflammation and ulceration. It involves a selection for viruses from the transmitting partner quasi-species with greater transmission fitness. And we still don't fully understand that because our in vitro assays don't seem to be translating into the same fitness that we're assaying in vivo. It selects for less glycosylated CCR5 using viruses, which likely take advantage of the alpha-4 beta-7 homing marker to target infected cells to the gut lymphoid tissue. In some cases, it selects for viruses with higher resistance to interferon, and that certainly seems to be the case for subtype C, but it's not apparent in the subtype C pairs that we've looked at, and certainly it was similar to what was reported by the Chauhan group. But it may be that this is dependent on the state of the transmitting partner at the time of transmission. I will say, though, that we now have the tools that we can, and it may be that we will be able to model transmission better in future studies using tissue explants and humanized mice. This expanding panel of authentic viruses from transmission pairs and acute infections that are now available will allow us to explore these possibilities. So I would just like to acknowledge the people that have really contributed a lot to some of the data that I've presented today. I've obviously reported on a lot of work done by other groups, but certainly these individuals have played a very important role. We could not do this without our colleagues in Zambia and Rwanda. I'm highlighting those in Zambia here. Cohorts that were initially started by Dr. Susan Allen. We have a wonderful collaboration with the International AIDS Vaccine Initiative and both Microsoft Research and colleagues at UAB. And I would like to thank Jonathan Carlson, Gustavo, Brandon, and Julie for their assistance in putting this together. And our studies are supported by a variety of funders. Particularly, we've got great support from the NIH, IAVI, USAID, the Canadian government, the UK government, as well as others. So I'd like to stop there. Thank you.